Okay, so you have to send me an email and I respond very quickly, as you will see. And we set up kind of office hours by Zoom, no office hours in person. And and it's unlimited. In person, we used to have 2 hours a week. And if you couldn't make it to my office at that time, that's it. And and not here. Here I give you office hours. Not everybody does that, but I do that. Office hours is unlimited here. So the mechanism is email, and then we set up and we use the same link, or you use the same link that you're using now for this lecture about being here in the lecture. Let me say that now that is the first time. Uh, the mechanism that the Zoom that I use is my own Zoom, and therefore I don't have some of the constraints of linking it to Brightspace because there are lots of problems there. So the Zoom is my own Zoom, and you can access it as long as you let me know so that I'll be there and open the Zoom for you. So this is how that part of communication works. The textbook, textbook is listed as required because it is. You will get tested on material here in the recorded lecture that you can view as many times as you like, whenever you like. So if that's enough for you, and people tell me that number of students that it is enough for them, so then you don't need the textbook. But sometimes to conceptualize, you can never answer a critical thinking question without understanding the concepts. Some of the questions are easy. They just test for factual information. And since it is an open book exam, it's easy for you to go to the lecture and kind of find the answer right there. Those are easy ones. Critical thinking questions require that you take A, you add B to it, and then you derive C. C is the answer. So that's the equation you got to work with. The textbook can help you with that. If you get it from my lectures, fine. Otherwise, you can read, and when you read, you think about it. And that's where the textbook is going to help you the most. So, having said that, let me move on to what is the mechanics of the course. And one thing is attendance. So, let me establish attendance, your attendance and mine right now, so we're clear until the end with this. There is no component of the final grade that is contributed by attendance. So in no way I can safely say that attendance is not required. Not totally true, because for the first 2 weeks or so, I need to check who's here and who's not. And if I don't hear from you, I assume you're here. But if you're not because you're going to drop it, let me know, please, because I need to report the verification of enrollment, and that has to do with your financial aid. Therefore, that's the only time where your attendance kind of comes into the picture. After that, for grading, uh, the lectures is recorded and it's available there for the whole semester. So uh, if you cannot be here, you're not really missing much. Only one thing will you be missing, and that is the opportunity to ask a question at the end of the lecture. And excuse me. And also, you can ask the question in the following time we meet in the virtual classroom. This is the virtual classroom. So in a way, you're not really missing anything because you have that option of asking the question later. So the the testing, that's something that concerns you a lot. Testing is going to be in bright space, not in Zoom. Zoom is only for the virtual classroom. Brightspace will take care of the testing. And that means if you are not familiar with Brightspace, take the tutorial. Definitely before testing, hopefully right now. so that you have that base covered when things get kind of more involved because other courses also come into the picture asking for your time. So the exam will consist of 20 multiple choice questions. There are different kinds of multiple choice. Some are critical thinking multiple choice. Those require more. So you will have 20 minutes for 20 questions, a minute per question. And these are the times here when the exam is going to be available, uh, depending on the section you're in. You're in section one, therefore, 8:30 to 9 a.m. If you come in after 9 a.m., Price Place closes the exam. You have no access. So you got to be there during this time when it's open, and then you will have your 20 minutes. So you have to manage your time because there is no backtracking and you get the questions one at a time. If you want to take 5 minutes to work on the question, it's up to you. But consider that you now will have less time for the remaining questions. And that's usually a problem. But it's a problem that you created for yourself. You got to be prepared enough to answer the questions, even the critical thinking questions, in 1 minute. So you really have to prepare, you really have to study. Now, what do you need when you come into the exam? Let's put it this way first. You don't lose anything. If you don't have a good computer and you don't have stable internet connection, and you're taking the lecture and it cuts you off, hey, it's not me. It's not my problem. It's kind of your problem, but it's not a problem because you get the recorded lecture available in Brightspace under content. Therefore, you're not really missing any critical material. That happens in the exam, and you lose the time that it takes you to come back into the exam. The exam is running. You're not there because the computer is not in good working order or you don't have a stable Wi-Fi connection. Those two are your responsibility. So if you don't have those two, you should not be taking a course online because you're going to have problems. And that's going to reflect in your grade. So this is quite important. So please don't forget this. Makeup exams, here are the conditions under which I can grant you a makeup exam. And in the old days, it all used to be in the domain of the professor. Now I need to have in the log of the course your doctor's note. In other words, the justification to be eligible for a makeup exam, because we can be audited, and then that's a violation on me because I'm not supposed to give you the exam. So this is what you need to get and provide. If you have a problem and you see a doctor, get a note and then you send it to me. You will also, the exam will be the same, 20 questions, 20 minutes, and we will set up the time separately, just you and me. And the grading, you know the grading, but just in case I have it here in the syllabus so that you don't have to kind of go and have to look that up. This is also your kind of choices, and it's good to be aware of your rights. Uh, Learning outcomes, this is a sampling of what you'll be able to do after you complete the course. Then what helps you here? Well, that's what I have here on the course tools. And of course, the textbook will help you. The lecture packets, which are posted in Brightspace even before the course begins. But those that you're going to see here, those don't help you very much because they have to be explained to you. So that's my job. So that's what I record and that's what I have available for you when I say the video of the lecture is in Brightspace. So these two kind of go together. You can, I don't recommend you study from the packets in Brightspace before the lecture. Those are there to kind of, if you want to go and see what's coming. Maybe you're the kind of person who likes to know what's coming, like me. When I was a student, I would go and check it out. And I say, okay, this is the number of slides we're doing today because I tell you in the syllabus the number of slides. And then when professor is talking, I'm kind of somewhat familiar, but you will see that I'd sometimes take 2 minutes to go through one slide. So that's the verbal component, and you got to know that, right? The exam comes from that slide, but the explained slide, not just the bare slide, which is what you see in 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 Brightspace. So two review sessions or discussions, you can ask all the questions you want before each exam. That is very helpful. If you don't attend, then I will go over the basic concepts for the entire part. Uh, but, and that is helpful. But if you're there asking questions, that's what really helps you. So you can ask questions or not. I always leave time for questions in every lecture because I'm required to do that. This is an online course and it is synchronous. So there's time for the lectures. So now the schedule of classes for us. And I provide you with as much information as possible to guide you. For example, the packets in Brightspace for the entire part one of the course. I tell you what packets are needed. I give you a chapter in the textbook here to kind of guide you, but I cannot have everything in the lecture covered in that chapter in the book. You gotta go to the index and look up the topics that are giving you problems. And then you can go to the page or pages and read them in the textbook. So that's how the textbook will help you. So that's provided here. Then there is the slides that I'm going to cover. Like today, what am I doing today here? Today I'm doing the syllabus, which I'm almost done with it. This is the last part of it. And then I cover homeostasis, uh, slides 1 to 15. There's more than that in the packet, as you can see here, but that's for the next time. Then the review, and then the exam in Brightspace. So exam will take place on March 3rd. When I say March, it already sounds warm because it begins to warm up outside at that time. And that's wonderful, right? So this is it. You have the whole thing here, lecture by lecture. And and that's actually all I have here. So I told you about your attendance at the beginning, the fact that nowhere here do you see what component of the attendance contributes what percentage to the final grade. It doesn't. So attendance is not required. I can say that. However, about me, I may have duties outside of the state, like I need to go to NIH to review grants. And that's a process that takes a couple of days. And it may happen that one of the lectures is there. I cannot review a grant in a discussion in a panel and at the same time talk for the lecture. Not you cannot be in two places at the same time. All right. So what do I do when that happens? Do we cancel the lecture? No. I let you know that I will not be in the virtual classroom. And then I go and I post the recorded lecture. So you can go at the time that you're supposed to be here, if that's how you manage your time, and access it and listen to the lecture. Or you can do it some other time when you are more comfortable in terms of managing your time. So this is really the last thing I have to say. So I'm ready to go to part one of the course, unless you have some question for me before I go. Okay, I'm not leaving. When I say I go, I'm just closing this and then I'm opening the first lecture packet. All right, so let's do that. And that would be right here. There we are. So first thing I do is I maximize and we're recording, so I don't have to record it again. So homeostasis involving fast cell-cell communication, that is nervous conduction. This is something the nervous system, the endocrine system uses as it brings about a response, a physiological response to a hormone. And that requires also that the hormone be taken, say, to the brain. And that's the nervous system. And there affects behavior, neural circuits that serve behavior. So large size in an organism, larger than a few cells, requires that you have a means of communicating. And that's what the nervous system does. It can happen by diffusion, but that is so slow that you cannot have anything complex going on. So we evolved the circulatory system to bring nutrients and remove waste to all tissues in the body that's required for large size, but also fast communication. And those are neurons. That's the brain, the central nervous system, also the peripheral. But we're going to focus first on the central nervous system. You can look up any of these concepts in a standard neuroscience textbook, any neuroscience textbook. That's why there's no chapter assignment for this. Uh, so let's take a look at how neuron to neuron, how that communication works, because it's critical here. Another thing that I didn't say before is the chat. The I can open the chat and keep the lecture going. So I look at the chat at the end of the lecture. I see that already one came up, but I'm not opening that because then we might even get disconnected from Zoom. So at the end, I look at that question and then I clarify it. Uh, so let's then begin. The central nervous system, which is part of the spinal cord and the brain, is in contact with the entire body, and the entire body feeds back to the brain. You will see in specific examples throughout the course how that works for some of these mechanisms that we're going to be studying. But it involves these stations here, all of which go in a feed forward mechanism, and they feed back to the brain, who knows what is the state of the body, because hormones also bring about responses that contribute to some kind of homeostasis. Sometimes the homeostasis happens only with the central nervous system, but it is also affected by the hormones, as you will learn in this course very nicely, very clearly. It's fascinating how that may have evolved. So then the brain itself. The thing with the brain, the human brain, in all other species, you have dorsal, which is above you, and then you have ventral, which is below to the ground, and that's it. But we rotated the brain so that we could stand on 2 feet and see what's in front of you. And that's important because you need to stay a certain distance from predators. And that's why this was required. And it makes survival better. So, but you have two coordinates that you have to look at because you have dorsal above, ventral below. In the brain itself, you have rostral. This is Latin for face, front. You have coral, Latin for back, and this is the back. But you also have, because the spinal cord is part of the central nervous system. It's continuous with the central nervous system. And that means that you will have a dorsal here, which is the back, like in all other animals, and then a ventral, which is the chest part or the abdominal part, which is it's down if you're on 2 feet and 4 feet. So that this is kind of what happened when we acquire this advantage where we can see what's in front of us with height, which gives you a better perspective, a better chance for survival. The brain has been mapped in 3 planes, and this is needed to do surgery, for example. That's how the surgeon knows where to go with this, and go in and take out a tumor, for example. So the coronal plane, as you can see here, it cuts the brain in 2 symmetrical planes. Imagine that you can go down and cut here, and then you remove this part and look at what's left, and you can see whatever you see on the right, you see on the left. But you don't know what's in front of you and what's behind. For that, you need another kind of sectioning. And that one is called the sagittal. It cuts from above or the top or the dorsal down to the ventral. The thing here is if you remove one half, you will see front to back, but you have no symmetry because you're cutting in a plane where no symmetry is possible. The other way of sectioning the brain is horizontal, the horizon. So you cut along this plane, say you remove the top, then you see what's underneath. You see it front to back, but you also have symmetry. You can see the left, you can see the right, but you don't know what's on top, right? Because you're cutting at that plane. But when you put the 3 together, try to in your brain do that. And then what do you see? Now you see the brain in 3D. And that's what you need to be able to go and know what part is contributing to whatever function you're looking at. So this is important also medical applications for surgery. Now we're going to look, now that we kind of looked at the whole brain, now we're going to look at what's inside. Well, inside you're going to find a lot of blood vessels and you're going to find cells. The first to be identified were neurons. Neurons come in different shapes in different parts of the brain. Sometimes a part of the brain has different kinds in terms of morphology, different kinds of neurons. And that's how it is. It's very heterogeneous. So some cells, like Purkinje cells in the cerebellum, they have a lot of dendrites because they receive a lot of input. This one, pyramidal cell, because the soma looks like a pyramid, so it's been called a pyramidal cell. This is found in many brain structures. It doesn't have something as elaborate as the dendritic tree that you see here. It's called the dendritic arbor in the Purkinje cell. But again, a field for receiving inputs, and then the axon for output. Some cells, like cells in the retina, they're very peculiar in the way they're set up. That's you can see here. Okay, so how do these cells then communicate? Because for the brain to participate in communication, you can see now that it means that the neurons have to communicate. And they use this by recruiting ions. So ions can be pumped to one side, and then they can be allowed to come in. What you have here is an ion transporter. And this is limited by diffusion. What do we mean by that? Well, no energy is required. If you're going to concentrate the ions on one side, say the outside, you need a pump to pump them out, and that requires energy. The passive transporters, like the ion transporters and the ion channels, they simply allow the ion to go to where it is not present in high concentration. In other words, diffusion controlled. The definition of diffusion is the movement of a substance from where it is in high concentration to low concentration until the system equilibrates, right? So these are passive. This is what they do. But you know already that neurons work by conducting an action potential, right? You learned that in introductory biology. So you know already that you got to have pumps and that you got to use a lot of energy. So this is what we're going to be looking at. The thing about these ion channels is that they can be regulated. They can be regulated by neurotransmitters. They can also be regulated by hormones, which could be coming from far away. So this we're going to see examples of this in detail. So essentially the neuron is looks like this. It has an area where inputs come in. You can see them here. Some of these are somatic, the others are dendritic. So these are the axon endings of other neurons, and they cause an action potential to be generated or initiated. So this is where the action potential initiates with information coming from another neuron. You can see this process magnified here. This is the presynaptic and this is the postsynaptic side of the neuron. So transmitter is released here, and then the transmitter causes channels to open or changing voltage by other mechanisms, and then that propagates through the axon in one direction. So this action potential then reaches the terminal and it makes synaptic contact with another neuron, let's say neuron B, or to use the proper term, the postsynaptic neuron. So this is the presynaptic neuron, and this is the axon terminal of the presynaptic neuron. The postsynaptic membrane has the receptor for the neurotransmitter. That's what is packaged in these vesicles here. And now you have transmission. So initiation, propagation, and transmission. That involves the neurotransmitter releasing to the synaptic cleft, and then that binds to its receptor on the postsynaptic membrane, which causes a change of voltage. This change of voltage is critical for the transmission. This is a real image. When you do these preparations, like this synapse appears to be hanging, it doesn't happen like that, right? It is very tight connection between the pre and postsynaptic, but it shows that what you see in these diagrams is coming from something that is real. This is from the cortex, and there can be other neurons. Very difficult to get a third neuron involved in the process. So the action potential, the initiation of the action potential. The action potential is initiated when the neurotransmitter that is released binds to its receptor and causes a change in voltage, which then propagates. So there are two kinds of changes in potential that you can measure here with a fine electrode, and that is the action potential. The action potential goes at the same speed, no matter how long the axon. Some axons are like a foot long and longer even in the periphery. The speed of conduction is unchanged in the axon during the propagation stage, and you can measure it and measure it here. This is what it looks like. You measure it here, and it still looks like the same. And then what is generated in the postsynaptic neuron is the same law: is constant speed, is all or none. It either propagates or it doesn't. But here in the synaptic cleft, there's something called synaptic potential. And that has a lot to do with how neurons kind of regulate themselves. Because one concept that comes into play here is frequency. That is, how many action potentials can arrive here in, say, 1 second. And that has to do with frequency. At high frequency, you get more. So the potential becomes bigger until it reaches threshold. When it reaches threshold, then the postsynaptic neuron changes its voltage and begins propagation of another action potential. So this synaptic potential is variable, and it allows for this neuron who has to be stimulated by another neuron here, right? It has to do with how much neurotransmitter is released in the synaptic cleft. If it's less than threshold, then it will not fire. So you gotta have the right frequency. That is the right amount of stimulation of this neuron to reach threshold here, and then another action potential happens. Meaning that different parts of the brain may be contributing to the contacts with this neuron at the dendrites. And if they all, because the brain commands all of them to kind of activate that neuron, then it's high frequency. And action potentials go on to the next neuron, one as soon as it is possible, one after the other. So we'll see what that means. A refractory period exists where the neuron has to do something before it can fire again. We'll see what that is shortly. So that's what I need to cover here. Look, very simple diagram. You look at it and I think you would know what you're looking at. But look how many concepts I introduced here and explained to you. This is what you gotta make sure you know and you understand to go into the exam. Because it's an open book. You can open this, that can probably help you prime your memory and bring things back. But the best way is to write it all down and study it. And then you'll be fine for the exam. Now we're going to see how you can build up a concentration of ions on one side. Essentially, what are these? They're arranged. These are the voltage gated sodium channels. There are many sodium channels in the nervous system, but this one here participating in propagation is the voltage sensitive type. We call it voltage gated. And you'll see in a minute what that does. So there's a pump here also, the sodium potassium pump, which uses energy to pump the sodium outside. So when the axon is not conducting, you have high concentration of sodium outside and very little inside. So there's a gradient. This is also a charge difference or potential because sodium is positively charged. So now you have mostly positive outside, mostly negative inside. This is what is needed to produce the action potential because when you go to your neuron here and you release enough neurotransmitter to cause this receptor to do the thing that it does. We're not going to talk about that now. And it then causes a change in voltage. So the sodium channels are voltage gated, voltage sensitive. What does it mean? It means that they change in conformation, they go from closed to open, sodium moves in. Now you have a dissipation of this gradient. You have also, you neutralize some of the negative charge on the inside, and then that causes more voltage changes to happen in the vicinity. So the voltage gating sodium channels now go and open in sequence like a domino. That's why they are arranged like a domino on a table. You push the first piece and it goes in sequence one after the other, no matter how long the sequence until the end. And if you measure the speed at any point along the first piece and the last, it's constant. The speed is constant. So it is for the action potential. We already mentioned that constant speed of conduction. So you also need then to have a pump that is going to restore sodium to the outside so that the neuron can fire again. That's called the refractory period, when these pumps are restoring the sodium gradient. And until that is restored, you cannot have another action potential generated. So this is the role of the voltage gated channel. in the axon. Then you get to the end, and when you get to the end, then you have a, a voltage-gated calcium channel. And what does it do? It opens when the action potential brings the change in voltage. It's essentially driven by changes in voltage. So it's kind of physics, isn't it? Yes, it is. But it's a kind of electrochemical physics. So this is kind of the biochemical part of it because you have this pump that will get the potassium restored at the terminal. But when this voltage gated calcium channel at the axon terminal opens, calcium moves in, and it is calcium that triggers the release of the neurotransmitter or the hormone, because you will see that some neurons release hormones even though they release everything like a neurotransmitter, like a typical neuron. So this, the important players here, the sodium potassium pump, and then the calcium is needed, the calcium pump is needed to bring the calcium out and stop the release of neurotransmitter. You don't want to release excessive amounts of neurotransmitter because this is a network. And if one part of the network is overactive, that's going to affect the entire network. So these things are very carefully regulated. So as you can see already, the brain uses a lot of energy because these things that happen constantly. Neurons don't rest. It's like the heart. They're always working. They rest between firings, like the heart rests between beats. So the vesicles fuse with the membrane. This mechanism is regulated by calcium, and kinase is activated by calcium, such as calcium calmodulin dependent protein kinase type 2. And transmitter is released, and you can see the role of the calcium, the voltage gated calcium channel, and then the effect on the postsynaptic, which changes the voltage. And then what happens here? Initiation of the action potential, and it will go like a domino to the axon terminal or for this postsynaptic neuron. The vesicles are recycled because when they fuse with the membrane, the amount of membrane surface increases, and that it won't work after a while. So you need to have also retrieval of the membrane happening constantly. And that's also an energy requiring process. Then you got to charge the vesicles, the empty vesicles, with neurotransmitter. And that is also an energy dependent process. How do you demonstrate that this recycling is taking place? There are a number of ways. In a neuroscience course, I would give you more than just this piece of evidence. This piece of evidence is kind of very old, very easy to do, but there's more subtle ways to demonstrate, some of which are biophysical, to demonstrate that the vesicles fuse with the membrane, that the transmitter is released, and that in the process of recycling, that is bringing back the membrane, you pick up substances which are going to be outside in the real brain, like growth factors, for example, which are needed. But you can demonstrate this process of picking up things in the process of recycling. So here you demonstrate recycling and also the ability to pick up something large. A growth factor is large. So is an enzyme called horseradish peroxidase. So it gets picked up, then you wash out the horseradish peroxidase or HRP. Then you give a dye called diaminobencidine, and this gets polymerized by this enzyme and forms a polymer that is brown in color. So you don't have to stain it or anything. It's all built into this simple experiment. The ones that involve microscopy, electron microscopy, the mechanisms that involve biophysical measurements, those are more complex. And we're not going to cover that here. But the concept of taking things into the neuron in the process of recycling, this experiment is demonstrated clearly, because then you can find the HRP inside, endopolymer forming inside, even when the neuron is not depolarized. So this is enough evidence for us of the process of membrane recycling. And in the process, you can pick up things that are there. Here you see now the process kind of broken into steps. We're not going to go into these steps. If you took biology 380 last semester with me, then you know the steps here, you know the proteins involved and how they bring about this choreography, and even how you store vesicles, which again is not relevant to us because here we want to look at endocrinology. And when you see this with neurotransmitters, you will see it here also with hormones. Some neurons release hormones, and they're true neurons. There's no synapse involved at the axon terminal, but the hormone is released and it does its thing when it goes through the circulatory system. So now we come, and this is the last slide for today. Retrograde signaling. And that means that the postsynaptic neuron, one of the responses of the postsynaptic neuron, you already saw it here, is to propagate the action potential. And that's what the postsynaptic neuron does. But they do other things also, one of which is retrograde signaling. For example, in this system, you have a GABA neuron here, presynaptic, and a neuron that has a GABA receptor, as it should, right? So GABA is released, synaptic cleft, GABA receptor, or here indicated as neurotransmitter. I'm using GABA as the example. And then this neuron will, GABA is inhibitory. It will not activate and change the voltage at the propagation site, the initiation site. But it can do that too if it is glutamate, which is a neurotransmitter that is stimulatory, unlike GABA that is inhibitory. So what happens? These neurons also have a mechanism to make an endocannabinoid. And these endocannabinoids are involved in a number of mechanisms. They're all over the brain. So this happens all over the brain. Needless to say, we don't know why this has to be all over the brain, but people say that is good for anxiety, that is good for depression, that is good for addiction, even though you might get addicted to this too, and so on. But the important thing is that when they are produced, they have effects all around because they diffuse, they're hydrophobic. They don't need a carrier. They go right through the membrane and then they affect the presynaptic neuron who has the endocannabinoid receptor type 1. And this will then cause an inhibition of calcium to get into the terminal. And what does that do? It kind of slows down neurotransmitter release because neurotransmitter release requires calcium. So it's a braking mechanism, a feedback which is negative. It requires the production of these endocannabinoids. Look at the structure, their chemical structure. They are hydrophobic. You cannot store them in a vesicle because they go right through the membrane. So that's why they also can get out of this cell and without needing a carrier and get into anything that is out there because they don't need a carrier. They just diffuse through the membrane. So this, in this example, is a braking mechanism to prevent excessive release of neurotransmitter. And that's important because I just told you, all of these neurons are part of a network that may have hundreds of neurons. So you don't want to overstimulate it because that is going to produce behavioral issues. So this is the last slide that we're going to cover today. And I will say something now that we're here almost at the end, and that is uh, I have practice questions. If you go to the Brightspace content site, you will see it there. You can open it and go over them before you come to the lecture. I cover them here at the end of each packet. So I think there are 4 questions. These are not on the exam. Exam is different. Different in the sense that they're not present. The pool in the exam is large, 120 questions, but these are not part of that pool. So The purpose of having that is to explain to you how I phrase, how I create the questions. Uh, that is what makes one question easy and the other one much more difficult. So that's what I'm preparing you for. The type of questions and the ones that are going to give you require a lot of time. So we're going to have those practice questions at the end of each packet throughout the course until the very end. I don't do that today because we're not done with this packet. I do that only when we come to the end. So if you have any questions for me, uh, I would like to answer them for you. Let me then go and see what's here in the chat.
Professor.
So then let me see.
Hello, Professor.
Who can see your messages, recording and message everyone? Uh, I don't know. I'll get to you. I'm sorry. I'm just trying to answer the question for this person. This went up at the beginning. If you cannot see anything here, it's at your end. I cannot help you with that. All right. Yes, you were asking a question. I'm sorry that I had to interrupt you. Go
For
ahead.
the horseradish peroxidase, what's the essence of it? The slide for that one.
What do you mean the essence? What it does?
Yes.
What it does is, uh, I cannot go back here with this. What it does is it converts diaminobenzidine, which is a monomer, into a polymer. The polymer has two properties: insoluble, so it doesn't get out, and it also picks up a color, brown. So you don't need to do staining to see who picked it up, and you always find it back in the vesicles inside the terminal because it was picked up by the process of membrane recycling. So it's indirect evidence of of this recycling of membrane.
So it's a evidence that there's always recycling at
That
the terminal.
yes.
Okay.
So if you picture it in your mind, in the brain there's no free space. The brain doesn't even store glycogen as energy. It relies on the liver to provide it in the bloodstream. So there's no room for anything. Now, that means that the synapse cannot grow. It must be maintained at that size because there's no place to accommodate it. So if it grows too much, it doesn't work. It loses the ability to work. So how do you keep it from growing into a large surface, a large size and having a lot of surface? You add membrane and you retrieve it immediately.
Okay.
That's what the recycling is doing, keeping the 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 membrane surface constant because there's no room to grow there. Okay.
Thank you.
Anybody else? Okay. So then I'm going to no questions. Then I'm going to sign off. I'm going to turn off the recording first.